channel Biopulses. Today we are going to start with a class that is Cell, the Unit of Life. This is the lecture number 5. Let's get started. Today we are going to discuss about mitochondria, chloroplast and ribosomes, cilia and flagella and some of the microbodies. Today's class is going to be a fantastic class because main organelle which are going to be asked for the NEET are going to be covered for today's class, and this session is going to be a bit lengthy, but be patient and listen whatever the concept I am going to explain today. Okay, now let's start with one of the very important organelle of the cell, that is the mitochondria, which is said to be a powerhouse of the cell because the mitochondria provides power because it produces the ATP. Okay, now, and what actually the main things which we are going to cover in this. Mitochondria is double membrane bound organelle. Which, why it is said to be a double membrane bound organelle? Because it is having the two membranes. Here you can see this one is the inner one and this one is the outer one. Okay, now found in eukaryotic cells. Mitochondria is found in eukaryotic cell, whereas it is absent in the prokaryotic cell. And it is also absent in RBCs of the mammals. Keep in mind this question is asked many of the times, where the mitochondria is absent. Mitochondria is absent in RBCs of mammals. RBCs is red blood corpuscles. Okay, now in RBCs of mammals the mitochondria is absent. And what is the main function? It produces what? ATP. ATP is an energy currency. Adenosine triphosphate we used to call. Okay, now the adenosine triphosphate, which is said to be an energy currency. Whatever the energy is produced in our body, that energy was produced by who? Mitochondria. In a cell there are many number of the mitochondria. The number of mitochondria varies. Okay, now mitochondria were discovered by Colliker. Who discovered the mitochondria? Colliker, he discovered in the year 1880 and named by Benda. The name mitochondria is given by Benda and it was discovered by Colliker. Let me show you detailed structure. Here it comes. See, here you can see this is the outer membrane. Okay, now, and this one is the inner one. Inner membrane is not smooth, it is going to be folded like this. Insidely it is going to be folded, and this folding is said to be what? Cristae. Okay, now this is the outer membrane and inner membrane is folded inwards. This inward infolding is said to be what? Cristae. And there will be a fluid like structure which is said to be a matrix. This is said to be what? Matrix. And what is the specification of the mitochondria? Mitochondria has its own DNA. Mitochondria has its own circular DNA and it has 70S type of ribosome. Okay, even being in a eukaryotic, the mitochondria has 70S type of ribosomes, because eukaryotes cell has 80S type of ribosome, but the matrix of the mitochondria has what? 70S type of the ribosome. Okay, now this is the inner one, inner membrane, which is going to be folded like this, and this infoldings of inner membrane is said to be what? Cristae. This one is said to be cristae, and the outer one is smooth. The number of mitochondria per cell is variable depending on the physiological activity of the cell. If the cell is going to be having involved more physiological activities, then that cell has number of the mitochondria will be more. If the cell is having very less amount of physiological activity, then the number of mitochondria will be very less. Okay, that means what? The number of mitochondria per cell depends upon how actually that cell is going to be work. Now this is what actually detail information about the mitochondria structure. Mitochondria has two membranes, just now I said, outer membrane and inner membrane. Outer membrane is smooth and permeable. It is chemically made up of 40% of lipid and 60% of the protein. Please keep in mind, many of the times question were arised based on this, the percentage of the lipid and then protein. The membrane of the mitochondria, outer membrane, keep in mind, outer membrane is smooth, permeable in the sense it is allowing everything inside, and 40% it is made up of lipid and 60% it is made up of protein. And whereas inner membrane is folded to form a cristae, just now I said, isn't it? I will show you once again. The inner membrane is folded to form a cristae and it is selectively permeable, whereas outer membrane is permeable to every ion, to every material, but whereas inner membrane is selectively permeable. It allows only selected things. Okay, now selectively permeable membrane having 80% of the protein and 20% of the lipids, is rich in cardiolipins. Which one is rich in cardiolipins? The inner membrane is rich in cardiolipins. Okay, see, outer membrane is made up of 40% lipid, inner membrane is made up of only 20% lipid. Outer membrane is made up of 60% of protein and inner one is made up of 80% of the proteins. Just be focused on this percentage. The two membranes are separated by a intermembrane space termed as perimitochondrial space. Let me show you. This is perimitochondrial space. See, this one is outer membrane, okay, and this one infoldings are there. No, that infolding is said to be cristae, or else inner membrane. Inner membrane shows infoldings. That infolding itself is called cristae. The in between these two there is a space, isn't it? That space is called as what? Perimitochondrial space. This space is called what? Perimitochondrial space. Within the inner membrane there is a large mitochondrial cavity filled with dense gel fluid called what? Mitochondrial matrix. Mitochondrial matrix is nothing but this one. This is the mitochondrial matrix. Whatever the bluish color is there, no, that's this mitochondrial matrix. The matrix contains high concentration of soluble protein, nucleotides, lipids and circular double-stranded DNA. Make this as a main point. Circular double-stranded DNA and 70S type of what? Ribosome. Here we will be having the circular DNA. Inside the matrix there is a presence of circular DNA with 70S type of the ribosome. Mitochondria contains 70S ribosomes for protein synthesis and has all three forms of the RNA. You will come to know the RNA's types. There are three types of RNA: rRNA, that is ribosomal RNA; mRNA, that is messenger RNA; tRNA, that is transfer RNA. These all three kind of RNA is present in mitochondria because it is having its own 70S ribosomes for the synthesis of what? Protein. The cristae and the inner face of inner membrane is studded with a numerous spherical knob-like protuberance called what? F1 particle or else oxysomes. See, when we are talking about the inner membrane will be having the studded spherical knob-like structure which is said to be a F1 particle, or else it is also said to be oxysome. This is the oxysome. Okay. Each oxysome is differentiated into base, stalk and then head piece. Base, stalk and then head piece. The headpiece contains the enzyme ATP synthesis, which brings about the oxidative phosphorylation. This is what actually ATP synthetase molecule, uh, one of the very important unit I can say. By this actually, this is the region where ATP is going to be formed. As you people know, mitochondria is said to be what? Powerhouse of the cell. Why it is said to be so? Because it is going to produce the ATP. Where actually it is present, the ATP is going to be formed. ATP is going to be formed in the ATP synthetase enzyme. This is the enzyme. It has F1 subunit and F0 subunit. By the help of F1 subunit, what happens? ATP is going to be formed. Mitochondria are self-duplicating organelles. It has the power of self-division. New mitochondria arise from the division of existing one. See, if the cell is in need of the more mitochondria, it is going to be duplicate itself. There is no need of cell division only. Okay, na? Mitochondria are believed to be bacterial endosymbionts of cells because mitochondria is believed that it is arise from the bacterial endosymbionts. Based on which criteria we are going to accept that one? Because they have their own nucleic acid, as the bacteria will be having their nucleic acid, and 70S ribosome we saw in bacteria, isn't it? Same kind of 70S ribosome we see in mitochondria. Even though mitochondria is present inside the cell of the eukaryotes, rather than the eukaryotes having this 80S ribosome, you people already know that. But even though the cell is having 80S, but mitochondria itself is having 70S ribosome. They show binary division. Mitochondria show what? Duplication in the form of what? Binary division, which is the main division of bacteria. Bacteria is also show what? Binary fission. That's what, based on these two main characteristics, we believe that bacteria, mitochondria is endosymbionts of what? Bacteria. Mitochondria depend upon nucleus and cytosol, that is cytoplasm, because of what? Raw materials. Therefore, structure and working of mitochondria is partially controlled by the cell nucleus and cytoplasm. Hence, mitochondria are actually semi-autonomous. Why semi? Semi in the sense half. Autonomous means automatically. Just now I said mitochondria is going to be divide itself by itself, isn't it? So it can divide automatically, but it is depend on the nucleus and then cytoplasm for the raw materials. What are the raw materials? Like other things which are going to be imported from the cytoplasm. That's what it is said to be what? Semi-autonomous. Okay, semi in the sense half. It is depend on the nucleus and then cytoplasm for its division. That's what it is said to be what? Semi-autonomous. Mitochondria inherited by maternally. Mitochondria inherited maternally in the sense what? Whatever the organism, that organism's mitochondria must came from its mother. Means what? If the baby boy or else the baby girl, in mammals if I talk, in humans if I talk, if the baby boy, baby girl is born, that baby's cells mitochondria were contributed by the mother. That's what mitochondria inherited by what? Maternally. Next, what is the function? Site of aerobic respiration. It shows what? Aerobic respiration. Aerobic respiration means taking of the oxygen and then giving out the energy. ATP synthesis, it is going to be oxidative phosphorylation. ATP synthesis is going to be happen in the mitochondria, and regulation of apoptosis. Apoptosis means programmed cell death. Heat production in brown fat. Brown fat, in the brown fat what happens? Heat production is going to take place by the mitochondria. Small sentence, that is mother gives power. Just now I said mitochondria came from our mother. So mother, whatever the power is supplied to the cell, and that cell is going to be supplying energy to the whole body to do any such kind of the things, that came from what? our mother. Next comes about the organelle. Let me once again explain the structure of this mitochondria slightly. Okay, this is the mitochondria, which is having two membranes, outer membrane and inner membrane. Inner membrane is not smooth, outer membrane is smooth, inner membrane is having infoldings, and that infolding is said to be what? Cristae. And the fluid like structure, which is said to be what? Matrix. Matrix is having the DNA, which is circular double stranded DNA, and the mitochondria has what? 70s type of the ribosomes. Mitochondria is made for ATP synthesis, and this ATP synthesis, these ATP synthase were present somewhere here. Okay, and this is F1 and F0 particle, which is said to be what? ATP synthetase enzyme. That's it. And the space between the outer and the inner membrane is called as what? Mitochondrial space. That's good. Now let's talk about the next organelle, one of the most important and then very interesting one, that is the plastids. Ernst Haeckel in the year 1865 gave the term plastid. The plastid term was given by Ernst Haeckel. Plastid are largest cell organelle. If we consider the cell organelles, among the cell organelles by the size, plastids are the largest one. These are involved in the formation and storage of soluble and insoluble carbohydrates. Plastids plastids are going to involve in the formation. They are going to produce and they are going to store also. What they are going to produce? They are going to produce the soluble carbohydrates, and they are going to store the soluble and insoluble carbohydrate. What actually the definition? Plastids are double membrane bound organelles. As like the mitochondria, these also having the double membrane system. Mitochondria is also having two membrane system. Same like that, plastid also having a double membrane system. Found in plant cells and algae. Plastid specially present in plant cell. It is absent in the animal cell. Okay, and also you can see in the algae. Involved in food synthesis, storage and pigmentation. Main thing is for the food synthesis, then next it is helpful for the storage, and next it is helpful for the pigmentation. Pigmentation in the sense coloring. They possess their own DNA and ribosome. As same like in the mitochondria, they also having their own double-stranded DNA and then 70s type of the ribosomes. It is also semi-autonomous. Same like mitochondria, the chloroplast also having the semi-autonomous. Semi-autonomous in the sense it can multiply itself, but it is going to be depend on the cytoplasm and the nucleus for the raw materials. There are types of the plastids based on the pigment. It is going to be classified into chloroplast, chromoplast and leucoplast. Chloro in the sense green color. Okay, now chloroplast, chromo in the sense color, leuco means colorless. Chloroplast, chromoplast and leucoplast. CCL, chloroplast, chromoplast and leucoplast. Chloroplast is for cooking food. That's what it is said to be, a kitchen house of the cell. And chromoplast is for coloring the organelle. For we get different varieties of colors in fruits, right? That color is due to what? Presence of the chromoplast in such kind of the cell. And leucoplast is for what? Storage. Keep in mind, this one is very important. Chloroplast is for cooking and chromoplast is for color. Leucoplast is for what? Storage. Here these all are. This is the chloroplast, which one you can see in a green color. Chloroplast will be green in color. Chromoplast will be different colors, okay, red, orange, etc, etc, etc. Next, leucoplast will be of again three types. I will explain later on. Etioplast is nothing but a precursor of chloroplast. Precursor of chloroplast means whatever the chloroplast is going to form, no, before that it will be like this. Now let's talk about one by one. First we will talk about the chloroplast. Chloroplast is helpful for the photosynthesis. Photo in the sense light, synthesis in the sense producing. With the help of light we are going to produce what? Food. That is going to occur where? In the plants. Okay, now green in color due to the chlorophyll. There will be a pigments inside the chlorophyll, chloroplast, that is said to be what? Chlorophyll pigment. It is found in leaf mesophyll cells. Inside the leaf there will be a presence of what? Chloroplast. And structure, if you talk, it is also made up of two membrane, which one is inner and then outer one. And fluid, let me show you. See, this one is inner membrane and this one is out. Both are smooth only, smooth and then round. But when we talk about the mitochondria outer membrane was smooth, but inner membrane was having the foldings, in foldings. Here there is no any such kind of the foldings. It is very simple. There will be a outer membrane and then inner membranes. Okay, whatever the fluid is there, no, this one, this entire thing, that fluid is called what? Stroma. And there will be a circular DNA and there will be a ribosome, which is 70S type of ribosome. Here you can see inside this stroma there are the disc-like structures are there, right? Yes, this disc-like structure are said to be what? Grana. This disc-like structure are said to be what? Grana. 1 grana, 2, 3, 4. These stack of grana. One second. Let me show you. See here. See, this is grana. Okay, this one thing, only one coin if I take, means a stack of coins you used to say, no, this only one thing if I say, that is said to be what? Thylakoid. Stack of thylakoid is called what? Grana. Only one coin is said to be what? Granum. Okay, stack of thylakoid is said to be what? Grana. Next, the fluid is said to be what? Stroma. The fluid is said to be stroma. What is the main function? Function of the chloroplast? Photosynthesis. Function of chloroplast what? Photosynthesis. The light reaction takes place in grana. This one. This is the grana. Stack of grana is said to be what? Thylakoid. Just now I said, right? So here 1, 2, 3, 4. The stack in the sense one after the above. Okay, grana, stack of grana is going to form thylakoid, and in this thylakoid one grana will be having the pigments which are going to be capture the light energy, and that light energy is going to be converted into chemical energy. Light reaction is going to take place here, and then dark reaction is going to take place in a stroma. Keep in mind this question may ask. Okay, now what kind of things are going to synthesize in the chloroplast? They are going to synthesize fatty acids and amino acids. Amino acids are going to produce what? Proteins. Fatty acids are going to produce what? Carbohydrates. So in brief, if I say chloroplast has two membranes, this is one membrane and this one is outer membrane, and it has a stack of grana which is said to be a thylakoid, and each granum has pigments, and that pigment is said to be what? Chlorophyll pigments. Not only chlorophyll, they are also having carotenoids, xanthophylls, many kind of the pigments are there. In later on in higher sections you are going to learn about those things. Okay, now what you should keep in mind here, The chloroplast is also having circular DNA as the mitochondria, and it also contains 70s type of ribosome, and this is the largest plastid. Chloroplast is the largest plastid and it is semi autonomous. Now let's move towards the next plast, that is chromoplast. Chromoplast is helpful for the pigmentation in the sense color. Okay, now it is colored plastid which show yellow, orange, red shades. Contain pigments like carotene. In carrot we will find carotene, xanthophyll, and it is found in fruits, flowers, aging of leaf. If you see, when leaf will be young, it shows green color because green color is due to the presence of chloroplast. When it is going to be age, means after some getting age, what happens, it is going to turn yellow, brown, like different colors. That is due to what? Presence of the chromoplast. Chloroplast is going to convert in the form of chromoplast. And what is the main function of the chromoplast? Chromoplast provides what? Colors. Why it is produce colors? To attract pollinators. Why it is going to attract the pollinators? See, if I say flower has color, flower color is due to the presence of chromoplast only. If the pollinator like insects are going to be attracted by the flower, that will be helpful for the pollination. Okay, help in the seed dispersal. Chromoplast will be help in the seed dispersal. Chromoplast can develop from chloroplast. Chloroplast, when it loses the chlorophyll pigment, it is going to be form in the form of what? Chromoplast. Chromoplast can develop from chloroplast. Next, last type, that is the leucoplast. Leucoplast is helpful for the storage. Chromoplast is helpful for the pigmentation. Chloroplast is helpful for the photosynthesis, whereas leucoplast is helpful for the storage. So it is the colorless plastid. Pigmented plastid is chromoplast. Green pigmented plastid is chloroplast. Colorless plastid is leucoplast. Where it is found? In roots, because root is not having the color, and tubers, and in some seeds. It is also having two membranes as it is a plastid. It is also having circular DNA, ribosomes, everything. But important thing about the leucoplast is its a type. There are three main important types of the leucoplast, that is said to be amyloplast, ileoplast, and alluroplast. Alluro word, wherever you will come to hear, it is representing what? Protein. Okay, now see what kind of, as I say, leucoplast is helpful for the storage. Each type of the leucoplast is helpful for the different kind of the storage material. Amyloplast store what? Starch. Ileoplast store what? Oils. And alluroplast store what? Proteins. Alluro in the sense what? Protein. Okay, see, this is amyloplast which stores starch, ileoplast which store oil, alluroplast which store protein. I hope this thing is clear to everyone. This is what about the plastids. Chloroplast helpful for the photosynthesis, chromoplast helpful for the pigmentation, leucoplast helpful for the storage. CCL. Chloroplast, chromoplast, and leucoplast. Now let's talk about one of the important organelle, that is ribosome. Already ribosome structure I explained during the structure of bacteria only, but then also some important things I am going to discuss in this session. So ribosome is said to be what? Protein factory, because it is going to help in production of what? Protein. Ribosomes are granular structures first observed by George Palade in the year 1953, and it is non-membranous organelle. It won't have what? Membrane only. It is only made up of rRNA and protein, found in all living cells. Ribosome will found in all living cells. 70s ribosome you can see in prokaryotes, 80s ribosomes you can see in eukaryotes. Exception. See, 70s ribosomes are present in prokaryotes, mitochondria and chloroplast. Just now I explained, isn't it? But whereas 80s eukaryotic cytoplasm, in 80s type of ribosome is present in eukaryotic cytoplasm. What is S actually? S is said to be a Swedborg unit or a sedimentation rate, we can say. What it is actually? It is indirectly is a measure of density and size. Keep in mind, already question were arised on this. What is S? Means S is the indirect measure of what? Density and then size. Each ribosome composed of two subunit. Each ribosome composed of what? Two subunit, that is larger and then smaller one. ATS has 60s subunit and then 40s. 60s is the larger one and then 40s is the smaller one, whereas 70s ribosome has 50s and then 30s. 50s subunit is larger one and then 30s subunit is smaller one. Don't get confused, don't add them, okay? 50 plus 30 you will get confused 80. No, it won't be like that. When they are going to be merged, no, that sedimentation, that size is going to be reduced. So that's what 50s and then 30s is gives 70s, whereas 60s larger and then 40s smaller subunit will give you what? 80s type of ribosome. Magnesium ion is essential for the binding the ribosome subunit. Whenever these subunits are going to be bound, no, for that binding, the magnesium ion is very essential. Next, what is function? Ribosome function is protein synthesis. The main function of the ribosome is protein synthesis, and there are free ribosomes, that is cytosolic protein. Bound ribosomes, that is they are going to bound in bound ribosomes, that is RER. When the ribosomes are going to be bound on rough endoplasmic reticulum, they are going to produce what? Secretory proteins. Okay, on smooth endoplasmic reticulum there is absence of ribosome. If the ribosome is free, then it is said to be cytosolic proteins, and if the ribosome is bound to the ER, that is endoplasmic reticulum, then it is said to be what? RER, which is helpful for the secretory protein. This is all about the ribosome which is said to be what? Protein factories. Next comes about cytoskeleton. Cytoskeleton, in the sense, inside the cell there will be a like filamentous proteinaceous structure which is present inside the cell or else inside the cytoplasm, collectively referred as what? Cytoskeleton. They are elaborated network, as like we the human body is having bones, right? We have bones. Due to the bone presence only we have stiff body structure. Same like that, due to the presence of cytoskeleton only, the cell has a firm structure. There are many types, mainly three types of cytoskeleton are there, that is microtubules, microfilaments and intermediate filaments. Microtubules helpful for the cell shape maintenance. Cell round, oval, rectangular, this maintenance of the shape is due to what? Microtubule, which is made up of tubulin protein. And microfilament is made up of actin, which is helpful for the cell movement. There will be a movement in between the cell. That movement will be takes place with the help of what? Microfilament. Intermediate filament, this is intracellular transport. Whatever the transport is going to takes place inside the cell, no, that transport is helpful by the what? Intermediate filament. And it is also helpful in the formation of what? Spindle formation during the cell division. You will come to know what is actually the function of spindle formation in the chapter, that is cell cycle. Okay. Next, microtubules are composed of protein, that is tubulin, which is about 25 nanometer. In plants, microtubules often found associated with cell wall. In cell wall, you can see the microtubules. During cell division, these microtubules form spindle fibers. Microfilaments, they are composed of what? Actin. And microtubules and microfilaments are part of what? Cytoskeleton base of what? Cell. Intermediate filaments. Intermediate filaments has these filaments form basket-like structure around the nucleus. These intermediate filaments will form basket-like structure around what? Nucleus. The next class comes about cilia and flagella. This is also very important. Cilia and flagella we discussed during the structure of bacteria only, but this is somewhat detailed structure. Cilia and flagella are hair-like outgrowth of the cell membrane. Cilia are small structure causing the movement of the cell or the surrounding fluid. Flagella are comparatively longer. Cilia and the flagella, when we compare, flagella is comparatively longer and responsible for the cell movement. The electron microscopic study of the cilium or a flagellum shows that they are covered with the plasma membrane. They are covered with what? Plasma membrane. Their core called as axoneme. They are covered with plasma membrane and their core is a central one. Possess a number of microtubules running parallel to a long axis. The axoneme usually has 9 doubled of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axoneme microtubule is referred to as a 9+2 array. 9+2 array of the cilia and flagella is, you can see in very firmly, 9+2. Cilia are small in size. When we talk about the difference, cilia is small in size and the flagella are long. Number of cilia per cell is very large. Few in number. Flagella will be very few in number. They take part in locomotion, attachment and feeding. Flagella involve only in the locomotion. Next comes about one of the important organelle of animal cell, that is centrosome. Centrosome and centriole. This overall thing is said to be centrosome, and 1 and 2. This is 2 centrioles. Centrosome is made up of 2 centrioles. Centrosome is a non-membranous organelle. It won't have any kind of the membrane. Found near the nucleus in animal cell. Centrosome only found in animal cell, and it is identical organelle also we can say, because if centrosome is present, then we consider that cell as an animal cell, and where it is going to present, it is going to present near the nucleus. It acts as a microtubule organizing center, MTOP of the cell. MTOP by microtubule organizing center. These all are the microtubules. Consist of 2 centrioles. This is 1 and this one is 2. Surrounded by a pericentriolar material, PCM. Pericentriolar material. This one is pericentriolar material. 2 centrioles are arranged perpendicular to each other. This is perpendicular. Okay, at right angle, perpendicular they will be. So these are the centrioles. Plays an important role in spindle fiber formation during the cell division. Organization of the microtubules helps in proper separation of the chromosome. Due to the spindle fiber formation, what happens? There will be a proper separation of the chromosomes. So centrosome function is what? The formation of what? Spindle fibers. That is the main function. Next comes about the microbodies. These are many membrane-bound minute vesicles contain various enzymes that are present in both plant and animal cell. First one is peroxisome or uricosomes, you can also call them. These are found in both plant and animal cell. Peroxisomes contain catalase enzyme. Which enzyme peroxisome contains? Peroxisome contains catalase enzyme, which is concerned with peroxidase metabolism, H2O2. This metabolism is due to the presence of metabolism takes place due to the presence of what? Catalase enzyme. Catalase degrade the H2O2 into water and oxygen. This is very important. H2O2 is going to be degraded by the help of which enzyme? Catalase enzyme. And what it is going to give? It is going to give in the form of water and oxygen. In plants, peroxisomes occur in cells of green tissues concerned with the photorespiration. That is said to be what? Glycolate pathway. Peroxisomes are also involved in both oxidation of fat acids, beta oxidation of fatty acid. Peroxisomes are involved in what? Beta oxidation of fatty acid. Next, one more microbody, that is the glyoxysomes. Glyoxysomes occurs only in plants, especially in fatty seeds like groundnuts and castor seeds. So the glyoxysomes are only present in plants, they are absent in animals. Glyoxysomes are considered as highly specialized peroxisomes. They are nothing but the highly specialized peroxisomes. Acid cycle takes place which converts fats into carbohydrates. This is important. Glyoxylate acid cycle takes place which converts what? Fats into carbohydrates. So after this we covered many very important organelles. Now let's talk about the questions, that is MCQs. So we discussed today about many of the organelles. First we discussed about mitochondria, which is said to be powerhouse of the cell. Next we discussed about plastid, in which we talked about CCL, that is chloroplast, chromoplast and leucoplast. Next we discussed about ribosome, which is said to be a protein factories. We discussed about cytoskeleton and its function, cilia and flagella, centrosome and centriole, and some of the microbodies. Now let's discuss about the questions. The inner membrane of mitochondria forms a number of infoldings. The number of infoldings called what? Cristae, cisternae, cristae, microtubules and mesosomes. Mesosomes are the infoldings of bacterial membrane, isn't it? So this is wrong. Microtubules are present in the cytoplasm. So this one is also wrong. So the correct answer is cristae. Cristae is the right answer. Next question. The type of ribosome found inside the mitochondria is inside the mitochondria. The type of ribosome, he is asking the type of ribosome present inside the mitochondria, which is 70S. 80S is present in cytoplasm. 90 and 60S ribosome is not there. So correct answer is 70S. Next. The plastids which store proteins, I said, whenever the word alluro, then it refers to what? Protein. So where is alluro? Yes, here it is. Alluroplast. First option itself is correct. Elaioplast, amyloplast and chromoplast. Elaioplast is for oil storage. Amyloplast is for fat storage, and chromoplast is for the coloring. So answer is alluroplast. Which of the following plastid is colored and contains carotenoids? Colored and contains what? Carotenoids. Color thing. Alluroplast is for storage of the protein. Elaioplast is for the storage of what? Oils. And amyloplast is for what? For the storage of fats and some of the carbohydrates. So chromoplast, it means what? Chromo word itself is said to be what? Colored. So the correct answer is chromoplast. Fifth one. Which type of plastid contains chlorophyll and responsible for photosynthesis? Which type of plastid contains chlorophyll and responsible for photosynthesis? First one itself, chloroplast. Chromoplast is for the coloring. Alluroplast is for storage. These two are for storage. So correct answer is chloroplast. Sixth question. Centrosome is an organelle containing two cylindrical structure called what? Centrosome is made up of two cylindrical structure. I said that is said to be centrioles. Seventh question. Microtubules are absent in mitochondria, centriole, flagella, spindle fiber. Microtubules are absent in mitochondria. Centriole has microtubules, flagella is also made up of microtubules, spindle fiber is also made up of microtubules. Grana are stacks of what? Thylakoid. One thylakoid, two thylakoid, three thylakoids, four thylakoids. Stack of thylakoid is said to be what? Grana. Each one is said to be what? Granum. Okay. Next. Axon with 9+2 microtubular arrangement occurs in cilia as well as flagella both. So answer will be both A and B. 10th question. Match the items given in column 1 with their role given in column 2. Choose the correct option. SER, smooth endoplasmic reticulum. Golgi apparatus. See, what you should do in exams, you must take care of time also. In the math following, if you are very confident in two of the options, any of the two of the options, you will get the right answer. For example, I can say Golgi apparatus, or else I can go with elaioplast, peroxisome. Okay, now see, elaioplast is helpful for what? Storage of oils and then fats. Yeast, where is yeast? See, yeast means this option is wrong and this option is also wrong. Next, any of the one. SER, smooth endoplasmic reticulum, synthesis what? Lipid. SER, synthesis what? Lipids. So A is 5. So right option will be A only. A is 5. B, we'll say all. Okay, Golgi apparatus. B is the excretion. Exactly. Cristae, increase the surface area. Peroxisome, excretion. So the right option is A. Means what? If you know any two options correctly, exactly, then you can go with the right option. It will helpful for your to maintain the time in exams. So from question number 11 onwards for your test series, I will give answer for 11th to 20th question in the next session, that is in the lecture number 6. So write down the questions. 11th one. A component of cytoskeleton is microtubule, bone, chitin, cartilage. 12th one. Match the following. Tonoplast, contractile vacuole, food vacuole, air vacuole. Contains digestive enzymes, store metabolic gases, excretion, transport of. Basal bodies are associated with the form of phragmoplast, cilia and flagella, cell plate, kinetochore. Which one of the following pair is not correctly matched? Cristae, the tubular structure formed by the foldings of the inner membrane of the mitochondria. Plasmodesmata, the membrane surrounding the vacuole in a plant. Grana, the membrane bound disc in chloroplast that contains chlorophyll and carotenoids. Medulla, layer between the adjacent cell walls in plants derived from cell plate. 15th one. Which one of the following combination is mismatched? Glycocalyx, maybe capsule or slime layer. Pili, reproduction. Cell wall protective, determines shape, prevents from bursting. Flagella, pili and fibulae, surface structure of the bacterial cell. Which enzyme highly concentrated in peroxisome is responsible for the rapid breakdown of the hydrogen peroxide into water and oxygen? We discussed about this, isn't it? I hope you people will answer this one very easily. 17. The centrosome typically consists of two centrioles lying perpendicular to each other. What is the characteristic microtubule organization in a mature centriole? Which of the following component of the cytoskeleton is responsible for maintaining cell shape and facilitating movement and is composed of actin? Which of the following function is not associated with microtubule? Not associated. 21. The triple arrangement of microtubule in a centriole is held together by a proteinaceous strands. The central part of the centriole is called what? Proteinaceous, and is called the answer will be in the next session. So this is an answer key, which is the questions are in the lecture number 4. So you can go through with those questions, and these are the answers. You can check them out. Last but not the least, today's motivational thought, that is, I am an autotroph of my own success. Means what? I don't wait for others to feed me knowledge. I seek out delight and then create my own growth. Just be autotroph. Autotroph in the sense, just feed yourself, feed your knowledge, and seek light and then create your own growth. For your own growth, feed your knowledge to yourself only. Okay. So this is for today. We will be ending the session here. Thank you. Subscribe my channel and hit bell icon for more videos notification. Thank you. Welcome back for the next session. Bye bye.